So good evening once again. The last part where we stopped was about identifying the different kinds of RNA. So we have the mRNA or the messenger RNA, the one that decodes the genetic sequence in the DNA inside the nucleus. And then later on it will carry the single strand RNA, which is an exact copy of one strand of the DNA also, to the ribosomes. And the rRNA, or what we call the ribosomal ribonucleic acid, will serve as a carrying enzyme. So the mRNA will be decoded inside this ribosomal RNA. So we have here, as you see in the picture, we have here the A site. This is the P site at the center. E site number 1 and 2 is A site. So A, it means accepting site, B, it means for the protein synthesis, and E means the exit site. So what are the purpose of these three subunits? So later on we will discuss during the protein synthesis. And now the last part is the tRNA, which means the transfer RNA, or commonly known as the translation RNA, because it translates the DNA, the sequence mRNA, into available protein or amino acid. So let's go now to the transcription. So we can see here in the picture that the double-stranded DNA will be unwinded. So see, this is the replication in the DNA synthesis. DNA replication, this is what we call the replication site. But during the RNA synthesis or the protein synthesis, this is a place where RNA polymerase takes place instead of the DNA polymerase during the DNA replication. So the RNA polymerase functions or acts almost the same with the DNA polymerase. So it will initiate in a process called initiation. It initiates the transcription process, and usually it starts with the promoter site, which is the theta box. So the RNA polymerase is the enzyme responsible for making copies of mRNA in the genes, as well as DNA and zips at the site of the gene that is needed. So in the transcription process, the single-stranded mRNA measuring from 5' to 3'. This is now the promoter sequence, which contains the theta box. Okay? And then there are some this is the upstream enhancer and the exons and the introns. So what's the difference between an exons and an introns? So the exons, keyword there is EX. It means these are the group of genes that will be expressed during the transcription process. While the introns, these are the group of genes that will be recycled or junked. It means they cannot be used. So this So again, the introns, they are the one being recycled or being junked. So they are not needed in the process, but hence they will be recycled back again. So only the exons. So how are the exons joined together? So the process during the process of cutting genes is what we're known as the RNA splicing. So splicing, splice from the root word splice, it means to cut. So how does our cells, or how does the RNA how are RNAs being cut? So we have this what we call the SNRPs, the small nuclear ribonucleic proteins responsible for making introns and exons. So they are the one who will choose which gene will be recycled or which gene will be connected to be expressed. For example, the yellow here expressed as the entrance. Well, the let's add here another one. While the blue are the entrance. So we want only the cell only wants the exons together. So they will try to cut between these exons and this entrance and take away the yellow part, which are the entrance. So the one that is responsible for the process is what we call the SNRPs, or the small nucleus ribonucleic proteins. So they are responsible for making the entrance and the exons. After they identify or mark the entrance and the exons, here comes the spliceosome. So the spliceosome splices the RNA strands, joining the good genes or the exons. So take away this. So I'm going to use this one. So you're going, back. Control Z. Okay, so they're going to take away this, and this, the yellow one, and join the blue sections. So the spliceosomes make it possible for the exons to be, joined together. So again, exons express good genes and introns are junk genes. They are cut out and they are recycled. So during the RNA splicing, this is how it looks like. The SNRPs will identify and mark where is the end of the exons and the beginning of the introns, and the SNRP will cut with the help of spliceosome. And then when they cut, the spliceosome will then take away a portion which is now called the junk gene or the introns, and these introns will go back again to the system and they will be recycled because they are also proteins. So here the mRNA structure looks like this: exon 1, exon 2, exon 3, but we only need exon 1, 2, and 3 to be to join, while the cell needs to take rid of the two introns in between. So during the RNA processing, the spliceosome will splice the RNA and join the three exons. As you see in the picture, the three exons are now joined together, and the RNA will then add the is going to add now or seal both ends of this strand. So the head or the 5' will be sealed with the 5' cap, and the 3' end will be sealed with the polyadenyl tail or the AAAA. It's also known as the polyadenyl tail. And they are considered as one of the untranslated regions. So the main purpose why the cell needs to put a cap and a tail in a single strand of RNA is because during the next process, if this strand contains the cap and the tail, like this is the example, this is the cap, this is the cap, and this is the tail. So during the transferring of the mRNA to the rRNA, it would be easy to get away or to get out from the nucleus going to the ribosomes because of the cap. So the cap helps in delivering the mRNA easily. And as a summary, this is the DNA. So the DNA will be translated via the mRNA, and the exons introns will be written off and then joined by the exon 1s, and then with a cap and with a tail. After you already have a single strand mRNA, which is composed of the expressed gene via the exons, transcriptions from the DNA is done. So the encoding in the DNA, it signals telling RNA polymerase to stop, and that is where transcription ends at that point. So you have now a single-stranded mRNA or the RNA, and that is coming one of the strand from the DNA, an exact copy. Okay. So with the help of the new structure of the mRNA, it is easy for it to go outside the cell nucleus, then travels right to the ribosomal RNA or the ribosomes. That's why RNA has its role now during the translation. So first, during the initiation of the promoter. Now let's go to elongation. And elongation is also known as the translation of proteins. Okay. For the translation of proteins, the chart here shows the different kinds of codes, different kinds of nitrogenous bases that codes for a single protein. Like for example, U, and U, it means phenyladenine, and U, and C is alanine. So these bases or nitrogenous bases codes for a particular protein. Example, the first letter is, if you have A, C, and G, as you see here, A, to find A is here. The second code is C, so C is the second, and the last code's for G. So it's either ACG, ACA, ACC, AC, and U, adenine, cytosine, and guanine. This codes for a protein called threonine. So threonine. So during the translation process, different three codes will be matched and interpret into a single protein, and this protein will link together via the peptide chain and creating and to be folded and creating a new structure of proteins, just like your red blood cells, your hemoglobins. They are an example of a folded protein. So as we see, this is now the rRNA. So the rRNA, we have the P site, the E site, and the A site. So the A site accepts, the P site will connect, and the E sites will release. So the single-stranded mRNA will undergo to the ribosomes. So just like this, the single-stranded RNA will undergo to the ribosomes, and these large ribosomal units will help in the translation process. Like for example, tRNA or the transfer RNA is just floating all around the cells together with the anticodons. So as we have discussed during the previews, the codons and the anticodons. So the tRNA are just floating everywhere with the three anticodons. So if the mRNA strands match, for example, C, G, and A, and the anticodon are G, C, and what do you think?